Exchanges. So, change the title, right? Change the title.
I changed it. It's only well, it's very slightly changed. Yeah, it's a sort of niche emergence, but yeah.
And now I'm going to be a person.
Okay, perfect. Yeah.
Buenos días y bienvenidos al seminario de investigación extraordinario de investigación del Instituto de Biología, perdón. Antes de iniciar, les recordamos que las puertas en ambos lados de la sala son salidas de emergencia. Si suena una alarma, usa la salida más cercana y por favor pongan sus teléfonos en modo silencio. Además, después del seminario, todos están invitados al patio para tomar un café o té con el ponente. Hoy tengo el grado de presentarles al Dr. Christian Taylor. Dr. Taylor cursó la licenciatura de Biología Aplicada y Bioquímica en la Universidad de Bath y el doctorado en la misma universidad bajo la supervisión del profesor Stuart Reynolds y el Dr. Alan Rayner. Desde 2006 es Associate Lecturer en the Open University y el Dr. Taylor tiene amplia experiencia docente, además de impartir materias en ciencias para escuelas secundarias. También enseña teoría de navegación y percusión de samba. El Dr. Taylor se encuentra de visita en México para colaborar con el Dr. Roger Guevara del INECOL sobre su línea de investigación de interacciones entre hongos y insectos. El título de su plática hoy es Surgimiento de nichos ecológicos en madera podrida debido a interacciones entre hongos y insectos. Bienvenidos, doctor.
Muchas gracias, Dave. Muchas gracias. It's a great honor to be here at UNAM, and I thank everyone for coming along today to hear my talk. As you can see, the slides are in Spanish, but I would like to remain speaking in English. I hope that's acceptable for everyone. My Spanish is not good enough yet to be able to keep up. So I'll start with a slide which may explain why I got interested in fungi and insects and creatures that live inside wood. I was taken to a park when I was a very young child in London, and I became fascinated by this statue, which is an old tree carved into many figures that exist within it. And I think that might have influenced perhaps some of the talk today. So when we look at a dead standing tree, we may consider the fact that the fungi community within the tree are the only microorganisms capable of decomposing the lignin within the tree and eventually rendering that tree back into soil. So the fungal component is vital for the decomposition of the wood. But inside that process is the ability for insects to dwell within the wood, and the particular aspect, although previous studies have looked at relationships between insects and fungi in the fruiting bodies of fungi, what I'm interested in, and the focus of the talk here, is the interactions between the insect community and the fungal community actually within the wood. So the mycelial component of the fungal organism as it relates to the insect community within the substrate of the decomposition process. So when we look at a fungal body, the actual organism itself, the mycelium, we can see the hyphal threads. We may be lucky enough to see a whole individual mycelium like this. Of course, when it's dry, the mycelium will be completely hidden from view because it will exist inside the xylem tubes, which actually make up the wood. So the hyphal threads that make up the body of a mycelium are just the right diameter to thread their way and ramify through a piece of wood. So it's quite possible to find a piece of wood and not see the fungal component within it on the forest floor or even high up in the canopy, where in fact 70% of the fungal activity occurs in a forest. Oops. So some of the classic examples are well known of interactions between insects and fungi. So the ambrosia beetle relationship is well categorized and studied, where the beetle will introduce a fungus into the wood, and within the gallery spaces, the insect larvae will grow, and you can see how they grow in size. They migrate towards the edges where the fungal mycelium is also living. So they're grazing on the fungal mycelium, which they introduce. And of course, we should remember the famous symbiotic relationships with the leafcutter ants and mycelia in tropical forests. And of course, by the same token, the termite fungi, which are well known to be living from the wood shavings, which the termites donate to the fungus organism inside the termite galleries. So when you look at a piece of wood on the outside, this is a piece of hazel Corylus avellana. You can see indications of the fungal community within it from looking at any recognizable fruiting bodies of the fungal organisms. So here, this is a Hypoxylon genus fungus, and you might not see, but I can point it out here. You can see another second fungus living in the same piece of wood. This is a bracket fungus of the Styrium genus. So it's possible for many fungi, in fact, a community of fungi, to live even within quite a small piece of wood like this one. And it's possible to cultivate fungi in the laboratory using 2% malt extract agar quite easily in petri dishes. So we can cultivate those fungi and subculture them. And also it's possible to introduce to those same petri dishes some of the obligate mycorvorous insects. So this is a Cecidomyidae, a peculiar form of dipteran fly larva in a family Cecidomyidae. And the interesting thing about this group of insects is that they do not form an adult form of the insect. So they exist only in the larval phase, in which they reproduce. And you can just see within this mother some little shapes within her body. Those are clonal offspring that are identical to herself in every way. And eventually she will burst open in a process called pidogenic birth, in which her daughters will migrate outwards from her body, and basically she reconfigures herself into smaller individuals. Underneath the aerial mycelium, you can just make out the shape of another insect underneath the petri dish surface. This is a look at the mouth part. You can just see a little horizontal mouth, which is ideal for eating hyphal threads. So this is an insect that has evolved only to live within fungal mycelia. So it's a good model organism to study the interactions with fungi. But in nature, when we look for the presence of these same insects in the field, what we find is they're in pieces of wood where there are many fungi, not just one fungus. So that presents an interesting possibility, because we can then culture the fungi perhaps in combination and look at the relationships with the insects then. The black lines that you see between individual fungal organisms basically are the boundaries between incompatible taxonomic groups. So where one fungus mycelium comes up against another fungus mycelium, it will produce this black line called spalting by carpenters, and by scientists it's called pseudosclerotial plate. Now, the interesting thing about these black lines is that they are chemically different from the mycelia on each side. They're actually produced by a process of melanization of organic compounds. And the other thing about the black lines, these PSP, I'll call them PSP lines, is that they are rigid and permanent, and they do not decompose. So the wood on each side will eventually decompose, but it'll leave this kind of buttress inside the decompositional architecture of the wood decay. So, so with that, I got interested in how that might create a kind of niche compartmentalization process. And of course, we know that carpenters have often used spalting for the beauty that it has. The PSPs are in wires, but many carpenters may not have been aware that actually these PSP lines, represent the boundaries between one fungus and another. So here we can see I've taken the bark off a piece of hazel and revealed a PSP line between Hymenocete coriegata, a basidiomycete fungus, and Hypoxylon fuscum, which is an ascomycete fungus. So very taxonomically different fungi living in the same wood. And you can see that two little cessid insects kind of resting on this PSP line. And if I did destructive sampling by exploring PSP lines and finding insects, these are the hemipupae of cessids. So cessids, when they become dry, can actually go into a kind of diapause state, and basically can, when they rehydrate later in the season, can then resume their normal life cycle of reproduction, which I described earlier. So, basically, we're looking at this interesting group of insects, the Cecidomyidae, with their pidogenic cycle. And we're considering that they exist within these, particularly between Hypoxylon fuscum and Volumina comedens, in these communities of fungi. And they seem to be particularly associated with the PSP. So the questions that I raised were, what is the effect of the influence of the interaction between the fungi on the insects? And then secondarily, what is how does the insect community change the interaction between the fungi? So this is the field woodland near Bath, where I was studying, log piles of Corylus avellana. So they had been cut and they were decomposing slowly. And I tracked them over 5 years to look at the development of the fungal and insect communities. Here we can see a close-up of Hypoxylon fuscum. And here's the other fungus, the Volumina comedens, which is the basidiomycete, which causes the bark to detach and roll back slightly, creating a little bit of surface area for the insects to shelter. But actually, here you can see some little insects under the bark, and you can just see them in the subcortical zone below the bark. Here's the life cycle of the insect. You can see I've crossed out the adult because they go through this pidogenic cycle. And the only way to identify them properly is to look at the microscopic surface patterning of these so-called creeping welts. So you can identify different species of cessid from the larval stage with a microscope. So growing them on culture, which were interactive mycelial cultures, here we can see the Hypoxylon fuscum, the ascomycete fungus, and down below is the Volumina comedens, the basidiomycete, and we can make out the larger sized mother larvae, very close to the interaction PSP line, which is replicated within the Petri dish. You can just make it out there. And also we can just make out some smaller daughter offspring, which are making their way through the mycelium, presumably grazing and feeding, but then perhaps, and this is the question I have, do they return later to rest on the PSP line? What is clear is that if you grow the interactive culture without any insects, So here is no insects at all. This is just the mycelia growing. The Vuliminia comedens would invade the territory of the Hypoxylon fuscum. So immediately it became clear that the insects were having an effect on the interaction between these two fungi in culture. So first of all, the presence of the insects on the PSP line became something to explore. Was it statistically significant? So I grew, first of all, I explored in the field and tried to map where I was finding these sessids. Very often on the PSP line was a very strong colony. And then in culture as well, in the laboratory. And you can see here the number of insects found on the PSP line in culture compared with the two pure mycelia on each side. So it does seem like we're finding more of the insects on the PSP zone. Now, it's important to point out that the PSP zone is not a source of food for the insects. So it's something else. There must be another reason why the insects are finding their way back there, but perhaps going off for feeding into the mycelia on each side of the PSP line. Also, I did a HPLC analysis of PSP to see if there was some unique chemical compounds associated with the PSP line, and indeed, that is the case. In fact, there was a very peculiar smell of the culture dishes when I opened the incubator, and the PSP actually had an aroma very similar to licorice, which is quite interesting. Anyway, so I started to grow cultured dishes with and without insects. And another effect became very clear, which is that the size of the PSP zone was enlarged when the insects were interacting with the colony. So we can see here with image analysis, in the dark blue represents the interactive cultures, where we've got the insects present, we can see much more extent of the PSP area in the culture dishes. The other interesting feature was to look at the trajectories of the routes the insects were taking, which were visible in the cultured mycelia. And we can see these wonderful loops where they seem to be going out and then returning to the PSP zone. So here's the Hypoxylon fuscum. This is the Vuliminia comedens, and then you can see these loop radii. So it's possible to measure those. So in the middle here, we can see the measured radii, the loop radii. And in the PSP zone, we can see that the loop radii, the average radius of these loops was much less. And there were sort of straighter lines, larger radius loops, foraging patterns in the mycelia on each side. The other thing which was different was that the speed that the insects were navigating through the mycelia was also altered by the interactive culture. So in the interactive culture, the newly born, just emerged, recently born young insects were moving much faster than in the pure culture. The other change was that actually in the pure culture, we can see here, now let's have to go up to the, you hear me still? Yeah, okay. So in the other thing is that the size of the insects altered. So in the interactive cultures, the insects actually grew to a larger size than they ever grew in the pure culture. And that was also very interesting. So there seem to be a number of different effects going on at the same time, comparing the pure cultures to the interactive cultures and the effects on the insects. As there was one other effect I didn't mention here, is the sessid number. The overall population reached a higher size in the interactive cultures. Here's the data again on the actual mean length, the body size of the insects between the interactive cultures and the monocultures. So what does this mean in the field? How can we put this together? Well, one thing that's very clear is if you look at PSP lines in nature, there's often associated with the PSP lines some physical cracking, micro fissures, and small cavities that open up in the wood alongside these PSP lines. So it might be that actually there's a kind of evolution of finding space within wood that is going on. And it's just that the insects are attracted to the chemicals, perhaps within the PSP, to find these cracks where they can fit physically inside the wood, perhaps. So to test that idea, I took slices of wood, of Corylus avellana, and I subjected the wood to 4 treatments. One was just water, the 2 pure cultures, Hypoxon-fuscum, Villaminicomedens, and then the interactive culture. And in the interactive culture, again, I was finding on the wood far greater populations after several weeks than in either of the other conditions. So when you slice a piece of wood across, you can sometimes notice these cavities that are opening up under the bark in the subcortical zone and associated with the presence of the PSP, the interaction between the fungi. So in a sort of schematic diagram, I tried to summarize some of that. And these red shapes are the different body sizes of the cessids. And when they give birth, the younger, smaller daughters can go into the smaller spaces, whereas the larger body-sized mothers, shortly destined to give birth, are perhaps confined to their quarters in the larger cavities associated with PSP. This is just an idea, but it's possible. So in summary, we have this interesting insect doing its pydogenic reproduction, associated with insects, with the fungi, the fungal interactions with the PSP. And it seems that, yes, that the insects are having an effect on expanding the PSP zone, and also that the interactions between the fungi are producing the right conditions for the insects. And what we sort of surmise, what we conclude from this, is that this insect is actually in the process of niche construction. So it's building it's actually making its niche bigger through its behavior with the interactive fungi. So what are the implications of this? Well, when you look at PSP within a woodland, you can see that it's persistent. It exists long after the wood on each side of the boundary between the fungi has gone, and you're left with a kind of crumbly honeycomb, a kind of honeycomb structure, which is brittle, waterproof, and full of cavities. You can see here there's a lot of insect frass. This is not cessid, this is something larger than insects now, but I'm looking at sort of the possibilities of over time, this becoming a kind of architecture which accommodates life in forests. And we can kind of postulate that as a forest grows, the cavity size and diversity of sizes grows with the forest, and we get more diversity of species over time building up. We know that every tree eventually becomes hollow, and we know also that a community of fungi is responsible for that. And we know that we can find lots of PSP within old hollow trees. And so it's possible, again, to create a kind of schematic between the living part of a tree and the uptake by roots on the other side, and this kind of sweet spot in the middle, where we get the maximum cavitation of wood associated with the legacy of fungal interaction causing PSP to build up, and so on. And so what I did is I put insect traps around pieces of wood that were where there was a presence of a fungal community inside the wood. So these are dead hazel poles, or Corylus avellana poles, quite thin, but with a community of insects. And I trapped all the insects that came out of a short section about 0.5 meter long. And I had 10 of these traps. And you can see the diversity of insects that emerges is astonishing. These are very small cessids up here, which are different because they do form an adult phase. So some cessids do form adults very occasionally. So it was interesting to catch those. We've got some some parasitic wasp, beetle, and some phorid fly diversity. But also, it's worth bearing in mind that the rarest beetle species in the United Kingdom, where I'm from, is the stag beetle. And here you can find, well, I found this one on a PSP zone. Obviously, it's just circumstantial. But this is within oak, Quercus robur, and you can see a large cavity there where the insect is associated with the PSP. And it's also interesting that one of the rarest species of beetle in Europe, the Glambricus phaeolaceus, the violet click beetle, only lives. The only place you'll find it is within these very large so-called veteran oak trees, which are hollow and full of PSP. And of course, many of the organisms that we love and try to conserve the most in the world live inside cavities and depend on these cavities. The cavity formation process, therefore, is becoming crucial for them. Perhaps you could argue that the lack of cavities is something of a conservation issue in many forests. And just to finish my talk then with one lovely example from Canada. So the male of this, the pileated woodpecker, actually has a courtship dance which involves inoculating fungi into saplings of trees. And generations later, that tree will have the right fungi softening the wood so that the cavity can be there when the next generations of pileated woodpeckers need a nest. But the nest is only occupied for a short time, for a few seasons, because the parasite load builds up and the pileated woodpeckers find a new nest. So the next creature that moves in is the flying squirrel, which is the keystone conservation species for eastern Canada and is a beautiful organism. But my question really is whether the fungi should be considered the keystone species here for creating the conditions and the cavities for both species. So that's the end of my official talk, but I just want to say that there is an international symposium for fungal research happening through the British Mycological Society website. So if you find the BMS website, you will be able to take part in October this year in a World Fungal Day extravaganza. It's completely free and it's organized by Professor Lynn Boddy from Cardiff University. So you'll be able to drop in and listen to it and even contribute to it if you'd like, if you have some research you'd like to contribute about fungi. I'm actually on the committee of the British Mycological Society Education Group. A couple of references. One is just about the insects, the peculiar insects that I introduced today, the cessids, and the other is about niche construction, which was a 2003 paper. And I'd look, sorry? Oh, thank you. Yes, sorry. Thank you very much. Yes. And then, thank you. And then just some acknowledgements. So just to say thank you for help at the University of Bath from Professor Stuart Reynolds and Alan Rayner, Dr. Roger Guevara, who's now at INICOL, but I met him when he was doing his research at University of Bath. So we were in the same laboratory for a few years. And then the funding for my research came from the Natural Environment Research Council. It's an English research organisation. British Mycological Society spun, which put me in touch with UNAM. So this is the Society for the Protection of Underground Networks, which is organised by Dr. Merlin Sheldrake, who's written the book Entangled Life, all about fungi, which has become a bestseller in Europe. And also, I'd just like to say thank you so much to UNAM, to the biology department, for allowing me to come here today and speak to you guys. And thank you so much for coming along today. That's the end of my talk. Thank you.
A ver si hay preguntas.
Thank you, Dr. Taylor, for your beautiful talk. I kind of suspected that you were going to talk about niche construction, but you surprised me in the way your example is relevant to that. So I have been studying from a philosophy of biology perspective what niche construction is and what niche construction theory means for a reform of evolutionary biology. So I really think that your example, I don't remember if it was actually included by Leyland and colleagues in the book, probably not, but I'm sure that if they know about it, they would celebrate it, no? So a specific question is, as you remember in the canonical scheme of niche construction theory, where you have environment and organisms interacting via niche construction, but also natural selection. What would be the role of selection in the examples that you have been studying? And also, which differences in niche construction do you find when you study, when you focus on the different organisms that participate in the assemblage, right? Because as we know, when niche construction has been progressing over the years, people, not only Leyland, Feldman, and Odlingsmith, but other people who have been publishing about this, have started to make differences between selective niche construction and ontogenetic niche construction, and they have come with many different classifications of different types of niche construction that can occur. So, can you tell us more about these, finer mechanisms that you are looking at in this beautiful example? Thank you very much, and congratulations.
Thank you. Thank you. Lovely, wonderful question. I'm not sure I can answer it very well, but I'll try. I think that the cessids have been selected recently to drop the adult phase of their life cycle. And we know this because some cessids under stressful conditions will actually leave the pydogenic cycle and go back into producing adults, but the adults are completely sterile. So it seems that this is an indication that this is a recent evolutionary change, and that it's ideal for the particular habitat where they're constructing their niche, if you like, with oh, I forgot I could speak here as well. So they can actually move in deeper within their territory where the mycelium is rich, where the food source is, because they're grazing on these mycelia. But then as they get bigger, they're expanding. They're getting restricted from their food because they're too big to fit through these micro fissures where the xylem is, where the mycelium is. So I think they've kind of recently developed this, I don't know how many million years, but it's a recent thing. And it's very interesting that in culture, very occasionally with stressful conditions, if the mycelium dries out, you will occasionally find these hemipupae, which are these dormant phases, and then occasionally the hemipupae will actually develop into male and female rather than actually. So they'll develop into two different, to develop pupae that develop adults that are of different genders but are sterile. So this is a very interesting group of organisms. Another thing that's quite interesting regarding the genetics is that the cessids have a very high ploidy number. So they have something like 40 odd ploidy number. And for that reason, cessids became an interesting group of organisms at the very early stage of looking at chromosome numbers and ploidy numbers. So back in the 1920s, there was some attempts to research this group of insects, but it wasn't related to field biology. It was just culturing them for their genetics. But it's quite interesting that they have a very high ploidy number. Maybe that's a way of getting around the asexual lack of diversity somehow. Yeah, I'm sorry I can't answer the question more specifically than that, because I need to also catch up on the research myself. I'm hoping to publish my papers from this quite soon I'm working with Stuart to finally publish my papers.
So you are going to do some research at INICOL. Do you have some specific questions related to this that you're going to be looking into in Mexico?
Yeah, one of the things I'm really interested in is whether the same PSP phenomena that's so easy to find in European forests is actually available here also in Latin America, whether we can find the same kind of distribution between cavities and PSP. So I'm going to be going out with Roger, looking at bits of wood, basically. Yeah.
I remember going a few times to the fields and people say, you know, you don't really see too many forests. We'll be in a national park, but they actually let the wood rot on the ground here. So I'd be curious to see where
no, absolutely. Leaving wood behind is such a useful thing for biodiversity. It's good to see changes in land management reflect that in terms of parks and gardens and so on. Yeah.
Hi, thank you for your talk. It was really pretty. My question is regarding the age of these interactions. Do we have any information? How old are these interactions? Seems to me that it would be important for conservation, knowing these interactions, which is, I guess, what you are doing, right? Documenting it.
Mm-hmm. Yeah, so when the log piles that I studied in the forest in England, the wood was quite fresh when it was cut, and the interactions actually started to develop, and it took about a year for the fungal interactions to become established naturally. So some of the fungi are latent within the wood even before it's cut, because fungi are often mycelia are dormant in a latent stage of colonization while the tree is still very healthy. And then as the tree becomes dysfunctional, so as various branches become shaded and less useful to the tree, the fungal community basically breaks dormancy and starts to grow, and that mycelium will then ramify through the wood as the wood basically dies.
So my question was more phylogenetically speaking, historically speaking, I guess these interactions are not just sort of opportunistic, they happen in other parts of the world.
Yeah, well,
the same species, right?
That's right. I mean, every time you have an interaction between an ascomycete fungus and a basidiomycete fungus in a piece of wood, you'll find these PSP lines very, very easy to observe in nature. And in terms of when we say how old they are, I mean, they establish as soon as the mycelia meet each other in the wood. So as soon as the mycelia become sensitive to the reciprocal metabolic exchange that mycelia give out, they start to produce this kind of skin. It's just a kind of skin between themselves. And in terms of the other question, the other part of how old is evolutionarily how old.
Right.
Yeah. So then I think they've been there, I mean, for as long as fungi have been living in trees. So, I mean, really.
So they occur in other lineages of Diptera that you know?
Sorry?
So they are in other lineages of Diptera that you know of?
So the fungi, we know that the interactions are there with producing PSP, and that's very easy to find. In terms of different insects that we find, although I studied a very early stage colonist in small pieces of wood, the cecid, I believe that the implications of the study are wider, and that there are many different insects that live inside wood that actually also are attracted perhaps or utilize the fact that there are these wonderful structures that are left behind after the fungi have finished their decomposition.
Okay. Thank you.
Bueno, vamos a agradecer a Dr. Christian Taylor y creo que hay café afuera. Thank you.
Well, I know how you get. Oh, yes. Sí, hay arepas más acá, ¿no?